Hello everyone, Mr. Linder here. In this video, I want to focus on the functionality of the norepinephrine neurotransmitter. In previous videos, I've talked about the acetylcholine neurotransmitter, and we went through how acetylcholine can be excitatory or inhibitory, depending upon which type of receptor that it binds to. It turns out norepinephrine has many different subtypes of receptors as well. And so norepinephrine can be both excitatory or inhibitory, depending upon which type of receptors it binds to. So I want to focus on norepinephrine, but I want to show you that there are many different types of neurotransmitters within our nervous system. So in future videos, we can do a summary of things like serotonin or dopamine or GABA or opioids, nitric oxide, etc. But in this video, I want to focus on the monoamine that we call norepinephrine. So norepinephrine actually interacts with several types of receptors. The norepinephrine neurotransmitter actually interacts with what we call adrenergic receptors. And there's two main subtypes of the norepinephrine adrenergic receptors. Those would be the alpha and the beta subtypes. What's interesting, though, is that the alpha receptors have different varieties. So there's alpha 1, alpha 2, and even the beta receptors have beta 1, beta 2, beta 3. So we'll be looking at the alpha and beta receptors and some of their variations. What we should note is that with the norepinephrine receptors, they are all G protein coupled receptors. So if you want to go back and look at the G protein functionality in more detail, you can take a look at the acetylcholine video. But G protein coupled receptors just simply means that the receptor interacts with a G protein before it goes on to exert its effects. The receptor locations are throughout the body, so you're going to find receptors in smooth muscle locations, cardiac muscle locations, glandular tissue, the central nervous system. You're going to find norepinephrine receptors all over the body. When you look at norepinephrine, and it is a monoamine, so what does that actually mean? It means that it's derived from a single amino acid. So the amino acid that gives rise to norepinephrine is the tyrosine amino acid. And we can use tyrosine to actually create several different types of neurotransmitters. Turns out tyrosine is used to produce norepinephrine, epinephrine, and dopamine. So although we're focusing on norepinephrine, you should note that you can get other neurotransmitters as well from this tyrosine amino acid. And this is not unique. We actually use tryptophan to produce serotonin. We use histidine to produce histamine. So a monoamine neurotransmitter is nothing new. We just use amino acids to produce this particular type of neurotransmitter. So we use tyrosine, and through enzymatic conversions, we can produce norepinephrine. We can also produce epinephrine. Essentially, epinephrine and norepinephrine bind to the same receptors, and so therefore they're going to have obviously similar functionality. It's just going to depend on, is it interacting with an alpha receptor or a beta receptor, and that will determine what kind of functionality we're looking at. The epinephrine and norepinephrine neurotransmitters can actually be deactivated. We don't want chemicals to always be active within the body, and so we have different pathways to deactivate the norepinephrine and epinephrine neurotransmitters. We use enzymes predominantly to deactivate the norepinephrine neurotransmitter. MAO stands for monoamine oxidase. This monoamine oxidase is in the presynaptic cells. And so when we reuptake norepinephrine, we can use monoamine oxidase to degrade the norepinephrine so that it's not functional. We also use in the postsynaptic cells COMT, that stands for catecholomethyltransferase. This is a postsynaptic enzyme that, again, can be used to inactivate through a degradation process the norepinephrine neurotransmitter. Clinically, we use this information. For example, we have what are called MAOIs. When you see I after something, typically you want to think inhibition. So if it's an MAOI, we're talking about monoamine oxidase inhibitors. Monoamine oxidase inhibitors allow us to increase the amount of norepinephrine that's in the synaptic clefts. That means it's going to persist longer, it's going to interact with receptors longer, and so it's going to exert its effects more often. This can actually be helpful, especially in the central nervous system, to have more epinephrine available for the postsynaptic cells. This can treat things like clinical depression. This can be used in, say, Parkinson's conditions. And so taking MAOIs can be beneficial for different scenarios clinically. As I said, norepinephrine can be used in different places in the body. So we see norepinephrine and even epinephrine, but we're going to focus mostly on norepinephrine. We see it released in both the peripheral nervous system and the central nervous system. In the peripheral nervous system, we tend to focus on the postganglionic sympathetic neurons. Those sympathetic neurons are going to innervate smooth muscle, cardiac muscle, glandular tissue, and so we're going to get those sympathetic responses in the body. So for example, norepinephrine can be used to increase blood pressure. That is a sympathetic response. In response to the norepinephrine chemical. But you should know that, again, epinephrine will do the same thing. So epinephrine is used to increase blood pressure. We're getting vasoconstriction of arterioles. So that's, again, affecting our overall pressure. We can dilate bronchioles in the respiratory system. You'll see that with a different subtype of the beta receptor. So we can treat asthma. We can treat hypotensive situations with epinephrine or norepinephrine because we can see those sympathetic kinds of effects. In the central nervous system, we tend to see norepinephrine being involved in sort of our general behavioral arousal. So just that sort of increase in cognitive functioning. This actually, though, what's interesting is it tends to explain why we see so much effects with amphetamine drugs or with cocaine because it is affecting these norepinephrine receptors in the central nervous system, and so we're getting that arousal effect that you see from these exogenous drugs. So they must be interacting with the same types of receptors. However, of course, exogenous drugs typically come with side effects. So you'll see cardiovascular damage, you'll see renal damage when people are using these drugs in a recreational kind of manner. In terms of the responses to adrenergic stimulation, so if we look at sort of the overall response to adrenergic stimulation, we want to focus on the idea that, again, norepinephrine is really a sympathetic kind of chemical, epinephrine and norepinephrine. So we see a lot of the sympathetic kinds of effects, especially out in the periphery. If the same neurotransmitter can have varying effects in the body, it's obviously because it's based on these different types of receptors. So I already mentioned that there's the alpha receptors, and then of course there's the beta receptors, but you'll notice that there's these various subtypes, alpha 1, alpha 2, beta 1, beta 2, beta 3. So we will see positive or negative effects throughout the body depending upon what type of receptor this chemical is actually binding to. So our two major classes of receptors are the alpha and beta variety, and then we see sort of the sub classifications: the alpha 1s, the beta 1s, the beta 2s, the alpha 2s, etc. The alpha receptors work differently. So they are all G protein coupled receptors, but you'll see that the alpha 1 works through a phospholipase C pathway, so I'll explain that in a little bit. You'll notice that the alpha 2 actually decreases cyclic AMP levels, which is different than what all of the beta receptors do. So all of the beta receptors work to increase cyclic AMP. And so we will look at the differences between them. You'll notice also the sensitivity. Some receptors are equally sensitive to epinephrine and norepinephrine, whereas sometimes norepinephrine is greater than epinephrine. Sometimes epinephrine is greater than norepinephrine. So again, their epinephrine and norepinephrine obviously are very similar. It just depends on what receptor subtype you're talking about and what kind of effect you're actually going to get. We'll look through the different varieties. The beta 3 is probably the less or least understood, but we do believe that it does have an effect on things like lipolysis, so the breakdown of fats, thermogenesis, so temperature regulation in our adipose tissue, and even brown fat, so white fat, brown fat activity. Clearly, if there's a lot of different subtypes to these receptors, you can use this knowledge clinically. So there have been a lot of pharmaceutical drugs that have been developed to interact with alpha and beta receptors so that we can get varying types of responses. Whether we want to increase blood pressure, decrease blood pressure, we want to bronchodilate to open up the respiratory system, and so forth and so forth. So there's a lot of pharmaceutical type things that you can take a look at. As I mentioned, all of these receptors work through the G protein coupled pathway. This is something I went through in more detail in the cholinergic receptor pathways, so when looking at acetylcholine. But just as a quick sort of summary of how a G protein works, a G protein coupled system is where you have a receptor. That receptor is tied to the G protein. The G protein comes with varying subunits, so alpha, beta, and gamma. And the alpha subunit will separate by itself, and then the beta gamma will stay attached. So when you activate a G protein, you actually split it into the alpha subunit and the beta gamma subunit. And then one of these subunits will move through the membrane and interact with either an ion channel, or they'll interact with an enzyme within the plasma membrane to exert its effects. So in this case, we're looking at this adenylate cyclase enzyme. Adenylate cyclase converts ATP into cyclic AMP. And so that's why you see a lot of the functionality with adrenergics is this idea of changing the concentration of cyclic AMP, either increasing cyclic AMP or decreasing cyclic AMP to therefore exert effects within the cell. So once you have cyclic AMP, you can go on, if you're increasing it, you can go on to activate kinases. Kinases are phosphorylators. So if you add phosphates to proteins within the cell, you essentially can go on and do work. So that's a quick little summary of how a G protein coupled receptor works. And alphas and betas use the G protein coupled mechanism. But they don't all use adenylate cyclase. Some examples are going to be using the phospholipase C example. But I'll start with the adenylate cyclase example. So I want to start with the beta adrenergic example. So in this example, what we have here is the norepinephrine neurotransmitter. So this is our neurotransmitter. Remember, though, that epinephrine can have similar functionality. So norepinephrine is binding to a receptor. And in this case, we're talking about a beta receptor. Specifically, we could be talking about a beta 1 or a beta 2 or a beta 3. So norepinephrine binding to a beta receptor, though, is going to have this similar functionality to it. So when norepinephrine binds to the beta receptor, what it's going to do is it's going to activate the G protein. So norepinephrine interacts with the beta receptor and it activates the G protein. When the G protein becomes active, the subunits separate. So in this image, it shows you the alpha subunit separating, but you should understand that you would still have the beta gamma within the membrane. That would be available in other scenarios. When you activate a G protein, you actually lose GDP, and the active subunit will pick up GTP. GTP. So the active subunit actually carries GTP energy. The active subunit, whether it's alpha or whether it's beta gamma, so in this example, we're following the alpha subunit. That alpha subunit is going to move through the plasma membrane and it's going to activate, like I said, either an ion channel or an enzyme. So in the case of the beta adrenergics, they interact with adenylate cyclase. So this is the adenylate cyclase enzyme in the plasma membrane. So what we have so far is norepinephrine gets released. It interacts with a beta receptor. It activates the G protein. And then we have the active subunit that's moving through the membrane and it's activating the enzyme adenylate cyclase. Once the adenylate cyclase is active, so for example, the alpha subunit binds to the adenylate cyclase, you're going to convert ATP that's in the cell into cyclic AMP. So the ATP gets converted into cyclic AMP by the adenylate cyclase enzyme. Once you have active cyclic AMP, then you can activate protein kinase. So what you can see here is we have inactive protein kinase within the cell. Once cyclic AMP comes along, and a lot of times you'll see cyclic AMP written like this, okay, little c AMP, the inactive protein kinase will become the active protein kinase. And again, protein kinases in general, okay, when you see kinases, these are phosphorylators. So they phosphorylate proteins. So what you're doing is you are essentially giving energy to proteins within the cell, or you could be affecting an ion channel, but you're doing some sort of work, okay, by activating the protein kinase. That kinase is going to turn on other proteins. It's going to give energy to something. It's going to affect an ion channel so that you can exert an effect on the postsynaptic cell. So norepinephrine binds to a beta receptor. It goes through the G protein process, and the purpose is to turn on protein kinases so that the cell can go and do further work. A different example of norepinephrine affecting a cell would be working through the alpha adrenergics. This example would actually be an alpha 1 example. The alpha 2s work differently, so we're going to focus on the alpha 1s. The way you get norepinephrine, so let's assume this yellow structure here is norepinephrine. Okay? So norepinephrine is binding to an alpha 1 receptor. The way you're going to exert your effect on the postsynaptic cell is again through a G protein. So you've got the alpha, beta, gamma subunits, and so one of the active subunits is going to move through the membrane. So for example, we could say that when norepinephrine binds to the alpha 1 receptor, we could get the beta gamma subunit. Okay? And of course, that would be carrying GTP, because anytime you activate a G protein, you lose GDP, and you pick up GTP. Okay? So we could say the beta gamma is the active subunit. The alpha would just hang back over here. And that active subunit is going to move through the membrane and it's going to interact with a protein in the plasma membrane. So in the case of an alpha-1 receptor, what we see happening is the activation of this phospholipase C. So we're activating this enzyme called PLC that's in the plasma membrane of the postsynaptic cell. So norepinephrine binds to an alpha-1 receptor, activates the G protein. The active subunit goes on to activate phospholipase C. So here we have in the key here, PLC stands for phospholipase C. Phospholipase C is going to work on a molecule. It's a membrane phospholipid known as PIP2. So PIP2. PIP2 stands for phosphatidylinositol diphosphate. And what you're going to do with phosphatidylinositol diphosphate is you're going to take that membrane phospholipid and you're going to enzymatically, so using phospholipase C, you're going to enzymatically convert it into IP3. IP3 stands for inositol triphosphate. If you forget, it's right here in the key. So inositol triphosphate. And you're also going to get this molecule, DAG. That stands for diacylglycerol. Okay. So PIP2, if you're looking at the functionality of phospholipase C is taking this membrane phospholipid, right? Membrane phospholipid. It's taking PIP2 and it's converting it into IP3 plus DAG. Okay, so you're getting two molecules for the price of one. You're taking PIP2 and making inositol triphosphate and diacylglycerol. Once you make inositol triphosphate and diacylglycerol, those molecules can go on to do work. So let's take a look at diacylglycerol. Diacylglycerol has the ability to activate a protein kinase. Okay? So PK, protein kinase, this just happens to be version C. You can have version A, so you can have different varieties. But again, the idea is that it activates protein kinase. Once you activate protein kinase, you know that kinases phosphorylate. Okay, so they add energy. Okay. So when you are activating a protein kinase and you're phosphorylating other proteins, essentially you're adding energy to them, and then that allows them to do work. So in this diagram, you can associate work with the idea of we get a cellular response. So phospholipase C creates diacylglycerol and IP3 from that PIP2 molecule. Diacylglycerol goes on to activate protein kinase. It's a phosphorylator. It allows the cell to do work. What about the inositol triphosphate? This particular molecule is going to interact with your ER. So it's going to interact with your endoplasmic reticulum. When IP3 binds to receptors on the endoplasmic reticulum, you're able to release calcium. So ERs, especially smooth ERs, are good at storing things. And so calcium is stored in endoplasmic reticulums. So when IP3 interacts with the ER, you're able to release calcium. That calcium, once it's in the cytosol, so now we have calcium in the cytosol, calcium is going to interact with a protein. It's not listed on this diagram, but I'm going to throw it in here for you. It's going to interact with something called calmodulin, very popular protein for calcium to interact with. You can remember Ca at the beginning of calmodulin goes with calcium. So calcium interacts with calmodulin, and that is in turn going to go on and alter the cellular activity. So IP3 triggers the release of calcium from the endoplasmic reticulum. Calcium interacts with calmodulin. Calmodulin and calcium go on to do work within the cell. So those are two different examples looking at beta receptor response and alpha receptor response to see how norepinephrine can exert its effect on postsynaptic cells. What kinds of responses, though, do you actually get? So let me mention alpha 2s really quickly, since that one's a different kind of response. I'm actually going to jump ahead here. So the alpha 2 response, let's go here. So on this particular diagram, let's look at the idea of the alpha 2. The alpha 2 receptors, so those that interact with norepinephrine or epinephrine, these tend to be located in presynaptic axon terminals. And when they're stimulated, they actually decrease cyclic AMP levels. Okay, so you notice right here, it shows sort of a blocking scenario on this diagram. So you're actually seeing a decrease in cyclic AMP levels when we're talking about alpha 2 functionality. It still works through a G protein, but it has an inhibitory process. So we actually see a decrease in cyclic AMP. Think of this as almost like negative feedback control, if you will, some form of negative feedback. So what you might see happening is that when norepinephrine interacts with these alpha 2s, you might see instead of a vasodilation in some areas of the body, you might see a vasoconstriction. But you also might see an inhibition in particular areas, especially the central nervous system. So for example, vascular smooth muscle tends to show vasoconstriction. So we actually tend to see smooth muscle contraction when you're out in the periphery. But if you look at alpha 2 agonists in the central nervous system, so if we look at CNS functionality with alpha 2 receptors, we tend to see the ability to lower blood pressure. So although you might see muscle contraction and vasoconstriction that might increase blood pressure in certain areas, if you affect the central nervous system, so the alpha 2 receptors in the CNS, we actually can see an overall lowering of blood pressure. So it's actually been shown that there are alpha 2 medications that affect the central nervous system and can actually be used to lower blood pressure throughout the body because it's actually decreasing overall sympathetic nervous system activity. So medications that work through alpha 2s actually can decrease your sympathetic control, and that actually can give you a decreased blood pressure response. They've also used some medications to interact with alpha 2s that actually are involved in like ADHD control. So there's some interesting pharmaceuticals out there for these alpha receptors. As a summary, how can we summarize, though, what some of the effects are for these alpha and beta receptors? So I want to go back to a table like this. This is a summary of selected adrenergic effects throughout the body. And the key here is to notice that you see norepinephrine affecting lots of locations: the eyes, the heart, the skin, the skeletal muscle, the lungs, the GI system. But it depends on what receptor it's interacting with. So are we interacting with alpha 1s, beta 1s, right? Beta 2s, okay? What type of receptor are we interacting with? And then we could even throw in beta 3s, and we can talk about alpha 2s like I just discussed, okay? So what are some of the overall kind of generalized effects that you see with these alpha and beta adrenergic receptors? So if you were going to give a generalized summary for alpha 1s, I would say alpha 1s consistently show contraction of smooth muscle. So we consistently see smooth muscle contraction when we're talking about alpha 1 receptor activity. With alpha 2, we see a variation. So sometimes we'll see vasoconstriction, okay? Other times we might see relaxation of the gastrointestinal tract. We might see decreased central nervous system effects, and so overall sympathetic nervous system decline. So depends on what location you're in. For beta 1s, when you think beta 1s, think increased heart rates, increased force of contraction of the heart. So think about increasing blood pressure and increasing the cardiac output of the heart. When you think about beta 2 receptors, I would focus on things like bronchial dilation, opening up the respiratory system, being able to breathe better. This would be vitally important for somebody that's in like an anaphylactic shock situation. You hear about epinephrine pins, right? So giving them an epinephrine shot, epinephrine, norepinephrine, opening up the lungs, helping them to breathe. Think of beta 2s when you think of asthma, inhaling a medication that's going to open the bronchioles, make it easier to breathe. For beta 3s, think of adipose tissue. So beta 3s, I would think of adipose tissue. Think about breaking down fats, lipolysis, thermogenesis. Those are good functionalities when you think about beta 3s. So how does this play in clinically? I've given you a few clinical examples. Beta blockers are a very famous type of pharmaceutical drug. Beta blockers can be used to interact with the beta receptors of the heart. This could, if you're inhibiting the beta receptors of the heart, this can reduce heart rate. This can reduce overall contractility, can actually help you to slow down the heart, can help you to reduce blood pressure. So beta blockers are a good example. You can also, though, have chemicals that interact with the beta 2 receptors, like I said, in the respiratory system. So you can affect the beta 2 receptors, and that can help with bronchial dilation. So in the lungs, open up the airways, make it easier to breathe. So it really just depends on what medication you're talking about and what selective subtype that medication is interacting with. So I hope you enjoyed taking a look at how adrenergic receptors function, whether it's an alpha adrenergic or a beta adrenergic. They can use different mechanisms to change the chemical concentration within cells and do work for that particular cell. So I hope this helps. Take care.